Thank you. Thank you very much for the invitation. And I'm going to come and talk to you today about a new approach to phase 2 and 3 evaluation of novel therapies, which we've been developing in the UK and we're about to launch. So you've seen this slide already today. We know that every cancer is unique and it's evolving. We also know that as they spread around the body, they are heterogeneous. What's the consequence of this? Well, we know that for many years we've been failing to make progress in some of our hard-to-treat cancers. The list is up there. We really are making no progress at all. A lot of that's about late diagnosis, but a lot of it's also about not really understanding the biology. So my field is colorectal cancer, and we have seen some improvements in outcome in colorectal cancer. Survival's doubled in the UK from about 25% to 50% in the last 30 years, but not a lot of that has been due to the activities of oncologists. A lot of it's been about earlier diagnosis and improved surgical technique. And frankly, our contribution has been very modest. And as we all know, improvements in chemotherapy have been slow. We were encouraged with irinotecan and oxaliplatin, and they have a very real place. Cetuximab and bevacizumab, over 10 years old, and really have they contributed much? They've changed our current practice, but the difference, the outcome, is relatively modest. And regorafenib, with its addition of just a few weeks, is really just adding to that list of misery, I think. We've heard this morning about our understanding of the carcinogenic pathways, and it's wonderful to add a third one to that list. When we look at the TCGA publication from last year, and we look at the mutations in colorectal cancer, first of all, the thing that came out was this distinction between the hypermutated group and the non-hypermutated group. And if you look at this hypermutated group, most of those are characterized by the mutations in the mismatch repair genes with which we're very familiar. And then pol epsilon here, which is also mutated, is a DNA polymerase, and there are mutations in the proofreading component of that. So as the DNA is polymerized, the proofreading is failing, so you get hypermutation. And that probably is, it may also be associated with a relatively good prognosis version of colorectal cancer, but there's work still underway on that. But what's going on in all of these other tumors? And this was the summary of the pathway alteration. And you can see 5 major pathways there. And again, just picking up from this morning's excellent talk, most of our therapies are directed at the RAS MAP kinase pathway or the PI3K signaling pathway. We're beginning to see agents in that may affect the P53 signaling pathway, particularly with the ATR inhibitor that's just coming into the clinic. TGF-beta signaling, very difficult to target, and Wnt signaling, also very difficult to target. So we're struggling with spreading the therapeutics across the range of pathways that are commonly mutated in colorectal cancer. So the last major trial that I ran was called the COIN trial, which looked at the addition of cetuximab to oxaliplatin-based therapy in metastatic colorectal cancer. And from that trial of 2,400 patients, we did an analysis, of nearly 2,000 patients. And so these are from clinical samples, all taken from the trial. 85% of these are from primary tumors, 15% from metastatic tumors. And you can see the mixture that there is, with the most common mutation being KRAS, about 9% BRAF, 12%, 13% PIK3CA, with a major overlap with KRAS, NRAS, a small group here. And scattered through, all these light blue ones are the ones with MSI, which are obviously not particularly linked to any particular well, more common in the BRAF, but also present without BRAF, leaving about 40% of patients with no mutation in any of those. So KRAS, we all know now that it's a biomarker of non-response to EGFR-targeted therapy. This is the first set of data which you're very familiar with. And here is the major trial, the first trial that showed in a randomized setting the way KRAS wild type was associated with some benefit from EGFR-targeted therapy. And the question is, where do we go now? So we have to recognize that every cancer is unique and evolving. But your approach, very interesting, was to take every individual patient and start again from scratch. I think that's going to be so hard to deliver in the clinic. And our approach is to try and work on biomarkers that stratify patients. So rather than personalized therapy, stratified medicine, where you're dividing patients up into groups so that you can learn about the prognosis of the group and potential therapies. We are getting better at understanding the pathways, and we need prospective biomarkers to enable us to stratify patients. And we have the opportunity to enrich populations so we can improve clinical efficacy. There are actually many new agents available, but very few successful trials. And as we've heard today, some trials enroll chemotherapy-resistant patients and all of the trouble and difficulty that results in. Many trials are inefficient. Trials that select the ALK mutation trials, for instance, screening 1,000 patients to get 40. How do we move forward? Well, we've been working over the last 15 years or so in the UK. We've had a very active colorectal cancer community. We've recruited nearly 10,000 patients into metastatic colorectal cancer trials. And in that process, we've been looking at a couple of things. One is intermittent therapy, which remains quite controversial, but we have shown that for about 75% of patients, the use of an interval following first-line chemotherapy is associated with no loss of overall survival. And we've also been looking at biomarker selection. And I'm going to talk to you about Focus 3 briefly, and then about Focus 4 down at the bottom here. So Focus 3 was a feasibility trial of molecular selection of therapy using KRAS, BRAF, TOPO1 in patients with metastatic colorectal cancer. And this was funded by the MRC and helped us to try and investigate this process. It's a complex trial, as all of these are. And if I can just talk you through this, we were looking at two sorts of biomarker. One was topoisomerase 1 immunohistochemistry, where we had some evidence from the previous trial that low topo1 patients could omit irinotecan with no loss of efficacy. And KRAS and BRAF wild type, and of course we know that if they're wild type, then they may benefit from cetuximab. And as we have no idea what to do with those who are mutant at this point, we thought we would use bevacizumab in that cohort. And our fourth hypothesis was that patients who had high topo1 might be those who would benefit from the addition of oxaliplatin. So we had 4 molecular types and 1 control arm, and 4 hypothesis-based investigational arms. And when I presented that to the national community, there was a sort of audible sigh. How on earth are we going to explain this to patients? And we worked with a couple of patients, Malcolm, who's an old patient of mine, and his wife, Jan. And we worked up a very simple approach to patient information. Stage 1, we were aware that we were talking to patients on the first day that they were diagnosed with metastatic colorectal cancer. And how could you explain that sort of trial to a patient when they've just heard that they've got incurable metastatic disease? So we drew up this diagram. We're going to do 2 tests on your cancer. It can either be positive or negative, and that will result in you being allocated to one of 4 types. Will you let us do that on your sample? Everybody said yes. Then a week later, when they came back, we talked them through the much more extensive information about the trial. Then when we had the results of the biomarker, we showed them the bit of the trial that was relevant to them. And then when they were randomized, they had all the full information. And that worked. And this was a questionnaire that we did to patients as they were going through that consent process, asking if they'd understood what was going on. And the purple is fully understood and the amber is mostly understood. So very high levels of understanding. And also very high levels of interest. The acceptance was very high. We accrued 240 patients to this trial within a year. Several of our investigators said this is the first trial they've ever had when everybody said yes. So it was successful. Our primary endpoint was to see whether we could get the sample from the day the patient gave consent in the clinic out of their path lab to a central laboratory to have the biomarker analyses back to the trials unit in 10 working days. Well, we only accomplished that in just over 2/3 of the patients, but we did accomplish it in 99% in 21 working days. And actually, randomization delay was far more often due to clinical issues than to biomarker issues. So this led us to believe that we can undertake complex prospective biomarker-driven randomized trials in the UK. And that's what we're going to do next. So what is Focus 4? So it's an integrated trial program that actually includes a number of clinical trials. It's totally adaptive, so we can change it as we go along if we need to. It exploits a window of opportunity to test novel agents in biomarker-selected patients in the interval following 4 months of first-line therapy. So it's moving away from testing novel agents at the end of the disease. We need to be moving these interventions nearer and nearer the front of the disease. And it's building on a statistical design, the multi-arm, multi-stage MAMS design, developed by colleagues Max Palmer and others at the MRC Trials Unit. And it's very cost and time efficient. And as I say, fully adaptable. So it looks like this. Patients in first-line metastatic disease have a diagnostic biopsy. They'll be consented and registered for the biomarker analysis. And then their tumour block will be sent to one of 2 central laboratories in Cardiff or Leeds, where they'll have mutations detected, mRNA measured for just for epiregulin for the subdivision of the all wild-type group, immunohistochemistry for mismatch repair, and P10. A big quality assurance process has been running between those 2 labs to make sure that they're measuring the same thing in the same way with the same protocols. That will then allocate patients into 4 molecularly defined cohorts, those with BRAF mutation, those with PIK3CA mutation or complete loss of P10 on immunohistochemistry, those with KRAS or NRAS mutation, those that have none of those 5 genes altered, and then a small group where leftovers where we have not been able to do the biomarker, which occurs in about 2%. And then within each of these groups, we'll randomize between a novel therapy and placebo in this interval. So we'll get a very clean readout of effect based on the primary endpoint of progression-free survival in the interval. So from our previous trials, we've shown that this is perfectly acceptable, have a break from therapy after 4 months in those patients who have normal platelets. And that's the opportunity we're going to use. And when they do progress, they just go straight back onto first-line therapy. We have not defined what is in first-line therapy, but we'll be stratifying patients within these comparisons for first-line therapy usage. So a number of different trials within this program. We're building in rebiopsy at randomization and rebiopsy on progression, not as mandatory, but as encouraged in all potential patients. So I'm going to talk you through 7 principles that we think are important as we think about this sort of trial. And the first one is this, that to evaluate multiple treatments and biomarkers in the same protocol. So we've been through the process of hunting for ALK mutation patients in non-small cell lung cancer, or just looking for the KRAS or the BRAF mutation patients. It's very wasteful. And this is a way of speaking to us in our standard clinical practice, where we see patients as colorectal cancer patients come to a particular practice. So it's patient-orientated, it's clinically orientated, and we should try, and it's efficient in terms of R&D management. The second principle is that we should investigate new treatments in the earliest and most likely responsive settings that are clinically feasible. So there are opportunities we have right up front. before a standard therapy commences. The neoadjuvant window is suitable in some settings. That's a great opportunity, which I'll mention again later. Possibly with some patients with non-aggressive metastatic colorectal cancer. Some studies have been done right up front before any other therapy. For most of our patients, they need to go on treatment fairly quickly. So this is the first possible window. And I think we need to think in each of the diseases we treat, is there a window? Or is our practice so driven by the desire to have people on treatment continuously, which patients hate? Is there an opportunity to actually make a window that is sustainable for patients and where we can do these studies? So what therapies are we using? Well, I'll be talking through these a little bit later, but we've tried to work with various different companies to identify agents which are relevant to these possibilities. The end one is just capecitabine. It's good to have a catch-all at the end, and I'll explain a bit more why. But the third principle here is that we must be using randomized evidence with a control group, because all of these different biomarker-defined cohorts have a different prognosis, so that each individual group has its own control. And that, again, is quite controversial. But in our previous study, you can see in the COIN trial, this is the survival of the BRAF mutants, much less good. The KRAS and NRAS run together, and the all wild type. And that's 8 months, that's 14 months, and that's 20 months. So 6 months difference between the prognosis and these different molecular cohorts with exactly the same treatment. So if we muddle them up or compare them with one another, we won't get clear answers about our outcomes. The multi-arm, multi-stage design splits up the analysis into these 4 separate groups. So if you like, you could call this a stage 2, a phase 2, and this a phase 3. But we're having an interim analysis for safety and lack of sufficient activity using PFS as the endpoint, and then a full analysis of PFS at stage 2 grade. And if we don't pass preset levels of activity, we throw it out and look for new cohorts, new biomarkers, new agents. If, however, we do pass this level of activity, then we move on within that biomarker cohort to a stage 3 analysis, but also we test the biomarker specificity in non-selected patients. So we're not making an assumption that the biomarker is driving the activity. So in the initial stages, assess each treatment in the presumptive biomarker in its subset, but without assuming in the design that this association will be confirmed in later stages. So if we look, for instance, at a targeted therapy, combination therapy for a KRAS-NRAS mutation group, if ever we find one, we'll need to prove whether or not whether that activity is limited to that cohort. So we'll use the cohort where we think there's best justification in the first instance, and then allow others in if it's showing activity in that best selected cohort. And this is what the statistics look like. So in the BRAF mutation, for instance, we're randomizing 2:1 to the active therapy. Here are the 4 stages, hazard ratio of 0.5. You can see the numbers and the time to accrue. So the power of this is that we're recruiting from every cancer hospital in the UK, and so we can recruit large numbers over time. And the principle here is you want rapid evaluation of each new treatment with flexibility of a phase 2 and phase 3 component, targeting a reasonably large treatment effect with discontinuation if no activity is shown. And this requires being right on top of the data, being able to analyze it frequently, having a very good statistics and trial management and data management team, which we have at the MRC Trials Unit. What about biomarker stratification? Well, these come in an order, and the order, of course, is somewhat random, but we are taking the BRAF group first as the first cohort. That's largely non-overlapping with KRAS, NRAS, and the others, so that makes sense. We're going to take the PIK3CA and PTEN status next, although there is overlap with KRAS, so that we will include the KRAS mutants in this cohort. then the KRAS and NRAS, which have not been covered further up, and if they're all clear, going on to the all wild type. Well, the principle here relating to this is that we can refine the biomarker. So how you actually measure the test will, of course, move on with technology. But also, you could bring in new biomarkers if you have a new hypothesis. The next principle of adaptation is that we could actually say, okay, we actually need to look at a subgroup on its own. So we may say that that group that has both PIK3CA and KRAS mutations, we need a slightly different combination to look at in that particular subgroup, and that can be introduced. Or we could go off in an entirely different therapeutic direction, allow the possibility to introduce a new biomarker and treatment into the overall trial program. How will we accomplish this? Well, over the last 10 or 15 years in the UK, there's been a major investment in research networks. So the NCRN is our National Cancer Research Network, which operates across the whole of the UK. The ECMC is the Experimental Cancer Medicine Centres. And so the recruitment will be coming from, there are 103 centres signed up for this, but the treatment with the complex novel combinations will just be at the Experimental Cancer Medicine Centres. So we'll be encouraging referral in for those early phase stages of the complex novel therapies. And we've worked a lot on how that will work. I think I've got a little bit to talk about the rationale of drug combination. I'll talk briefly, we've had it mentioned already this morning, about the V600E. There's the BRAF mutation. We know that it's meant to signal down the MEK pathway. If we inhibit it, we thought it would work, but it didn't in colorectal cancer, so they've tried it with the MEK combination. And then the lovely work from the lab here showing the EGFR signaling and the current phase 1, which Emil talked about this morning. And we've got commitment from GSK that they'll put this triple combination into the trial when it comes through phase 1. PIK3CA mutant P10 loss, it looks from Xenograft Experiments that a combination therapy is more effective, and we are looking for that combination at the moment from a partner company. It's been difficult, as you're all aware, these combinations of novel agents is being really difficult to get activity and safety together. So it is being slow. And similarly with KRAS mutation, we're wanting to inhibit both pathways, and we're waiting for seeing whether the schedule will be feasible. Again, GSK have committed to supporting this if they can get a tolerable schedule with their AKT and MEK combination, which is currently on trial. The one that's ready to go immediately is the all wild type, where we have the AZD HER1-3 inhibitor. So the other thing is about how do you really gain information as you go through this. So I've talked about rebiopsying. We're certainly used to collecting germline DNA on all of our studies, and we'll also be collecting circulating tumor DNA sequentially through this program, and I'm just trying to get some money to do all the analyses for these. But I think just one slide, not about this trial, but about how we could use this sort of model to bring forward treatment trials into the earlier stage of disease. So at the Gray Institute, we're working on radiotherapy-based studies. And so take an example of locally advanced rectal cancer, where, again, you could undertake imaging and biomarker analysis, incorporating a wider range of analyses. We know that hypoxia is a very important predictor of radioresistance. We've got very strong evidence in the laboratory that PI3K inhibition makes hypoxia go away in animal models. Does it work in the clinic? Well, that's something that we're beginning to test in phase 1 trials at the moment, which we would want to lead on to combination studies. We know that patients have, about 22% of patients have ATM loss in colorectal cancer. Many of them have P53 mutations, so they've got loss of that DNA signaling pathway and ATR inhibition in addition to chemoradiotherapy. So again, using biomarkers to subdivide the clinical population, and maybe on the basis of Richard's talk earlier on, the DDRD deficient, adding in an extra DNA damaging agent. Is this fairy tales? Well, we're already doing this in phase 3, so we'll be bringing that back into a biomarker-driven cohort. We're working on a trial very much like this in pancreas cancer. In fact, using nelfinavir. So that's a repurposed antiretroviral drug which has the AKT inhibitory activity. And that makes hypoxia go away in preclinical models, and we're using it in the clinic. And this is under review as a concept with one of the companies. So I think this sort of approach is the sort of thing that is generalizable. We're in fact quite, in one way pleased, in one way frustrated, that Roche have taken the design of Focus 4 and applying it into their new colorectal development plans. So it is doable, it's deliverable. And I thought that perhaps after this morning, we might actually think about this as a group who have very early propensity to metastasis. So this sort of thing needs a massive team, and we have in the UK a great team, Richard Wilson, who works with Paddy Johnson in Belfast, a group of clinical investigators who each lead different stratas of the trial, biomarker specialists, the clinical trials unit, the research networks, the funders who've found it very difficult to get their heads round, but Cancer Research UK recognise this as a flagship approach to stratified medicine trials, and the government funding as well. The pharmaceutical companies are prepared to collaborate so long as they're in separate molecular cohorts. Working with the higher education institutions is quite interesting, and then a translational science grouping which is being built at the moment. So they're the people who have made this all possible. And I'll just leave you with the 7 principles that we've worked on, which have been accepted for publication in the JCO just now. So thanks for your time.
Thank you very much, Tim. Are there any questions? Come on.
Kamara Zeile, VU Medisch Centrum. A lot of tumors have shown mutations in P53, but why is it so difficult to target these cells?
Well, because we haven't had agents, I think, that can really attack that weakness in the cell. And I think that the possibility of, I mean, the P53 is obviously a crucial signaling molecule on the DNA damage response and controlling apoptosis and cell cycle. So how can you target it? Well, either by hitting the myriad of downstream pathways, which is very difficult because there are so many of them. But I think the possibility of ATR inhibition, so ATR is on the parallel pathway, ATM through to P53. It may be that that will be one way, but you know, I don't have a good answer for that.
Okay, thank you.
I make it brief. It's interesting talk and very important items. I would like to know your opinion on there was a review recently by Barbara Parsons in which she gave evidence that actually there is no wild-type KRAS. It's a matter of your technique. And if you're doing DNA sequencing, you will measure 50%, but if you go further, you can find in all colon cancers mutated KRAS. It's a matter of time. The lesser mutated KRAS, the longer the duration of your remission. Could you comment on that?
Yes,
because I think it's a very important observation.
So I think there are 2 levels of that. First is that in the current standard practice, we don't look at all of the KRAS mutations that are easily detectable. So we just look at the codons 12 and 13, and it's perfectly easy to measure codon 61, 146. The deeper you go, the more you find. Certainly, it's very easy to push the KRAS mutation status up to 50%, and if you're going very deep, no doubt it'll get higher and higher. I mean, I think you raised the point that was raised this morning, which is within the mutation range that there is within a cancer, every resistance mechanism is already present. And I think the big question that we have Paddy raised it earlier today, are we actually chasing rabbits down rabbit holes, I think was his words. And in metastatic disease, has the game already been lost? And therefore, should we be moving all of this further up the disease? And that's why I put up that slide about neoadjuvant studies, because actually that's going to be our focus in Oxford moving forward, is not in metastatic disease, but focusing on using the biological pathway understanding, the genetic analyses, the functional imaging, to try and refine and personalize therapy in non-metastatic disease, to give us the chance of actually maybe curing more patients rather than just prolonging survival. And I think that's where our priorities should be, to early diagnosis and much more effective frontline therapy.